Hello, this is the CHEM271 review session. This episode we will be going over the first practice midterm for the first midterm exam. So the first question on this practice midterm is to draw the structure of the peptide GRIP with all stereochemistry and at pH 7. So the first thing we have to do is determine what amino acids make up the peptide GRIP. So the G here stands for glycine. The R stands for arginine. The I stands for isoleucine. And the P stands for proline. So the first thing I will do for this problem is to draw the peptide backbone with the correct stereochemistry, and then I'll add in the R groups afterwards. So I'm going to start with the N terminus on this side. So this will have NH3 plus at pH 7, and then I will draw my first alpha carbon. The stereochemistry on the R group will be pointing out of the page here, and this will be labeled as R1, and then the next is the carbonyl, then another nitrogen, then the next R group on an alpha carbon. This one will be pointing into the page. This will be R2, and then we just keep repeating the stereochemistry pattern until we have all of our amino acids drawn. So the next one, R3 out of the page, then carbonyl, nitrogen, and then R4 into the page, and then ending on the carboxyl group. This will have a negative charge at pH 7, right? So now that we have this drawn, now we can add in our R groups. So I'm going to erase the R groups. We can draw these in. So the first amino acid here is glycine. Glycine is the simplest amino acid. It's just a hydrogen. The next one is arginine. So arginine is 3 carbons in a row. And then there is a nitrogen group at the end. So 1, 2, 3, and then nitrogen with an H, and then this is connected to 2 NH2s in resonance with a double bond. This one here will have a positive charge. This is a basic amino acid because it has a positive charge at pH 7, so it will be a proton acceptor. The isoleucine, now for the stereochemistry for this, what we have to do is we have to put the hydrogen in front. We can keep the long carbon chain, CH3 with a CH2 in the middle, and then the one CH3 can go into the page. So doing this gives us the L amino acid, or the S enantiomer, because with the hydrogen facing out of the page, we can rank the groups 1, 2, 3. If we see that, they're going clockwise, and if we reverse that to have the hydrogen going into the page, that gives us counterclockwise S enantiomer or L amino acid. Lastly, we have proline. So proline's interesting because it is a cyclic amino acid, meaning that the R group is actually connected to the nitrogen in the backbone. So in order to get this, there are 3 carbons here that make up the ring, and then we have this connected to the nitrogen, and then these connect back to the alpha carbon. Okay, but this part right here is still going into the page. And that gives us the peptide GRIP, the correct stereochemistry, and with the correct charges at pH 7. Question 2 is, why are glycine residues found in beta turns? So glycine, as we remember, the R group is just a hydrogen. So it's very small. So small R group. The small R group is important because that means that this R group allows for flexibility. And it's flexible due to the low degree of steric interactions. So what this means of steric interactions. So this essentially means that when you have an R group that's this small, it can sort of get in the way of other R groups without causing a large increase in energy that would make it unfavorable. So it's more favorable because it's small, it's not getting in the way, versus if you have like a tryptophan and a phenylalanine that are sort of clashing, that would cause much higher steric interaction, higher in energy, it would be less favorable. All right, so the next question, question 3, why do no proteins have phi-psi angles of positive 120, negative 120? So to think about this, we can use a Ramachandran plot. So what this looks like is on the x-axis, we have the phi, and then on the y-axis, we have the psi. Okay? And then up here, we have positive 180 degrees. Down here, negative 180 degrees. Here we have 0. Again, down here on the x-axis, negative 180, 0, and positive 180. All right. So essentially, in this quadrant, we have beta sheets. So this is a negative phi, positive psi. Here we have a left-handed alpha helix. This tends to be pretty rare. So this is positive phi, positive psi. And then here we have the right-handed alpha helix. These are more common. This is a negative phi, negative psi. Okay? But here in the problem, we have a positive phi and a negative psi. So that lands us in this quadrant. This quadrant is not sterically favored. So we don't see these in nature because this is not allowed. So for an exam, I would recommend memorizing this. To actually explain this, we have to look at the structure of a dipeptide here. So looking at two amide planes, what is an amide plane? So an amide plane is when we have an alpha carbon and we have two amino acids. So this one is connected to a nitrogen with a hydrogen on this side and a carbonyl on this side. Okay, this alpha carbon has an R group going into the page and a hydrogen coming out of the page. So on the alpha carbon, that's what we have. This is connected to another carbonyl, and then this is connected to another alpha carbon over here. On this side, this is connected to a nitrogen, and this is connected to an alpha carbon. Okay, so here with this dipeptide, we can draw two amide planes. So an amide plane extends from alpha carbon to a carbonyl, and then the carbonyl to another alpha carbon, and we do the same thing with the hydrogen of the NH. Okay, so that gives us one amide plane. Now we can draw a second amide plane over here in exactly the same way. Okay, and now we can see that this dipeptide, all the rotations are essentially going to be rotations of these amide planes. Why is there no rotation around this bond? That's due to resonance. So this double bond here can come down and make a double bond here. So this bond is inflexible due to its potential to be a double bond. All right, and then we call rotation around this here to be our phi, and this one here is psi. Okay, now for the exam, you do not need to understand how these things rotate, but for example, I am going to show on this little model. So this is sort of the base, what I've drawn here. This is positive 180, positive 180. If we look at this, this is sort of up here, so it's favorable, okay? So why is this favored? It's because the R group is sort of facing out of the way of these amide planes, so there isn't steric interactions. But if we rotate this such that we have this configuration, positive 120, negative 120, what we end up getting is something like this, where the R group is in plane with the carbonyl and the hydrogen here. So drawing quickly what that looks like. So we have the R group here in the plane, And then here, this carbonyl, nitrogen, alpha carbon, and then on this side, nitrogen, carbonyl, alpha carbon. So what we can see here is that this R group is in the plane with this carbonyl and this hydrogen, and this is at positive 120, and this is negative 120 over here. Okay, so you don't need to actually know the rotations. You don't need to actually be able to rotate these in your head on an exam. What you do need to know is this, and that in this quadrant, this is not favored because we have steric interactions, so interactions due to these things being close to each other in space. This is unfavorable, this is high energy, and thus this will not be favored. And we can look again at the model to see that these are all kind of in the plane, they're all close together. So therefore, it'll be less favorable because they want to be about as far apart as possible. They're more far apart if this R group is rotated such that it is facing out of the plane. All right, question 4 has us drawing 2 parallel beta strands. So I am going to draw the parallel, but I will also draw the anti-parallel just for comparison. So the parallel beta strands mean we have 2 beta strands going in the same direction, like this. And this is only possible if we have some kind of a connecting loop that can connect the front end of one to the back end of another and giving us this parallel structure. So in the parallel beta strands, we have 2 going in the same direction, so we can draw 2 identical ones right on top of each other. So it looks something like this. Alpha carbon. The alpha carbons have R groups and hydrogens attached to them. So okay, like that. And then we can draw a second one that is completely identical right beneath it. See alpha, R, hydrogen, NH. All right, so now we have to draw where the hydrogen bonds occur. So hydrogen bonds occur between dipoles. So, and one of the dipoles has to have a hydrogen connected to an electronegative atom. So if we look at this nitrogen-hydrogen bond, what happens here is the nitrogen is more electronegative than the hydrogen. So it will pull electrons away from the hydrogen. This will give the hydrogen a partial positive charge and the nitrogen a partial negative charge. So we indicate it with this kind of arrow, and the little cross at the top indicates the positive atom. So in this case, the hydrogen will be positive, the nitrogen will be negative. If we look here at this carbonyl, we can do the same thing, except the oxygen is more electronegative than the carbon. So what will happen is the carbon will become more positively charged, the oxygen more negatively charged. And now what we can see here is we have a negatively charged oxygen in proximity with this positively charged hydrogen, and these will do a hydrogen bond as indicated by this dashed line. So this is a hydrogen bond. Okay, and the hydrogen bonds are what are holding these beta strands together. So if we were drawing all of them, there are only 2. Here, there's one here, there's one here. These are diagonal, which means these are longer H bonds because they are diagonal, and this makes them a bit weaker. So these bonds are not as strong as in a anti-parallel set of beta strands. So these ones are parallel. Now over on this side, just for comparison, we'll draw anti-parallel. All right, so here we'll draw 2 that are sort of staggered, so they are not going to be identical. So here what we'll have something like that. So we'll have a carbonyl facing down, the nitrogen facing down, the alpha carbon here, and then we will not necessarily completely stagger them, but instead we will draw them in sort of opposite directions. So, and then So this one here will actually be a nitrogen. Okay. So these are sort of flipped in this direction and then flipped in the other direction as well. So then this oxygen will be facing up. Then we'll have a nitrogen here. All right, and then these ones have the R group and the hydrogen. And these ones are going to be in opposite directions. So, right? So these are staggered and they're also in opposite directions such that the strands can flow directly from one into the other, like that. Okay, so here the hydrogen bonds are a little bit more clear. They are short and they are straight. So they are connecting this carbonyl to this hydrogen with the nitrogen here, and this nitrogen-hydrogen to this carbonyl. So these bonds are stronger because they have better alignment. They are shorter H bonds. All right. One last thing to note here is that in both of these configurations, the R groups are going to be facing out of the beta sheets. So in this case, if we look at this, the R groups will sort of be facing in or out of the page. They will not be facing in the middle of this plane, so that reduces steric clashes as well and makes beta strands either parallel or anti-parallel favorable. All right, now on to question 5. Question 5 is, how does urea impact protein structure? So urea has the ability to hydrogen bond, all right? And it will hydrogen bond to areas of polarized charge within a protein. So to areas of charge in a protein. And what this will do is this will disrupt non-covalent interactions in the protein. So by non-covalent interactions, I mean it will disrupt hydrogen bonds in the secondary structure, or it can interact all sorts of different types of bonds in the tertiary structure. So this can include hydrogen bonds, the hydrophobic effect, van der Waals interactions, all sorts of things in the tertiary structure. So however, the type of bond that it cannot disrupt is covalent bonds. So this means it cannot disrupt those disulfide bridges that connect tertiary structures and also sometimes quaternary structures together. So it's only disrupting the secondary and tertiary structures, and specifically just the non-covalent interactions like hydrogen bonds or dipole-dipole interactions. Okay, so that's how it's disrupting protein structure. It's sort of taking apart those secondary and tertiary structures. All right, so question 6. Why are hydrophobic protein residues normally found on the interior of soluble proteins? This is because they do not interact well with water. So and this is due to the principle of like dissolves like. So polar things tend to dissolve polar things. Non-polar things like hydrophobic amino acid residues tend not to mix with water. They tend to rather mix with other hydrophobic things. This is the hydrophobic effect. So they will naturally sort of separate due to the fact that these hydrophobic residues do not want to interact with water. They'd much rather interact with themselves. It is more energetically favorable. So they do not interact with water, so they go to the interior of the protein to shield themselves from the aqueous environment. So interior them from aqueous environment. Okay. And then this is essentially the hydrophobic effect, as I said. Again, these nonpolar hydrophobic residues would rather be with each other in the interior of the protein, shielded from the water, than they would interacting with the water itself. So on the inside, they're shielded, and the hydrophobic effect, as well as some van der Waals interactions, instantaneous dipoles, those can also hold it together. All right? And then the next question is, where in membrane proteins are hydrophobic residues typically found? So in a normal protein, they are found, that is water soluble, they are found on the interior, but a hydrophobic residue in a membrane protein would be found on the outside of the protein. And this is because on the outside of a membrane protein, they can interact with hydrophobic membrane lipids. So again, these hydrophobic nonpolar residues want to be with another hydrophobic thing, such as a membrane lipid, thus they will be on the outside and they will not be exposed to water. All right. The next question is a bioinformatics question. So we are going to compare the following protein sequences, sequence 1 and sequence 2. We're going to assign 10 points per identity and subtract 25 points per gap. So let's compare the sequences. So whenever we see an amino acid line up, we will circle it. So there's one here, we have a P and a P, and then we scan through, and then here we notice a gap. So there are 7 amino acids missing here. However, this counts as one continuous gap. This is not 7 gaps because there are 7 amino acids missing. This is one gap because it is one continuous gap. So this counts as one gap, okay? Next up here, we see the D here aligns, the F, the L, and the K, okay? So here what we see is we have 5 identities, which then we multiply that by 10, right? We're giving 10 points per identity, and then we are subtracting 25 points per gap. We said we have one gap, and then we multiply that by 25. So what we get here is 50 minus 25, which gives us a score of 25 points for this alignment. Now the second part of this question is asking, how would scrambling sequence 1 help to determine if these two sequences are related? So what we would do here is we would scramble sequence 1, so that means kind of randomly shuffle all the amino acids, and then we determine the alignment score each time shuffled. So scramble sequence, and then determine the alignment score each time we scramble, okay? And then we will plot these scores. All right? So what we want to do then is compare the distribution of the random alignment scores to the actual alignment score. So what this would look like is a graph sort of like this. Here, on the bottom here, we have the alignment score. Let me move this up a bit. All right, so down here we have the alignment score. And then on this axis, we have the number of alignments. Number of alignments, alignment score. Okay? So if we plot all these scrambled sequences, we should get some kind of a normal distribution of alignment scores like that. We know that this is a good alignment if we see our alignment score here. So let's say this is plus 25. This is the alignment score that we got. This is all maybe like, let's say 10 to 1. Okay, if we see that our alignment score is significantly higher than the alignment scores from scrambling the sequence, we know that it is unlikely that this alignment occurred by chance, and that there is more likely an evolutionary explanation as to why these two sequences are related, why they share so many identities, okay? So if there is no overlap, likely alignment occurred by chance. Right? And there's probably some other evolutionary relationship going on here that would explain this. Right? All right, question 8. This one is about hemoglobin and myoglobin. So the first part is to draw and label the binding curves for both myoglobin and hemoglobin. So looking at the graph we are given, we have gamma on the y-axis, which is our fractional saturation. On the x-axis, we have PO2. This is pressure in torr, the partial pressure of oxygen specifically. So we are going to draw the two curves. So we have that curve. This curve is for myoglobin. And then we also have a curve that's more sigmoidal, like this, for hemoglobin. Okay? And then up here, this is the point at which both of these proteins are saturated. So from this, we can label their P50 score. So right here is 50% fractional saturation. So the P50 for myoglobin is very low. I didn't draw it particularly well, but P50 is supposed to be about 2 torr for myoglobin, while here it is about 26 torr for hemoglobin. So P50 equals 26 torr for hemoglobin. I didn't scale it particularly well, but the point still gets across. So the difference between these is myoglobin is just a normal hyperbolic curve. Hemoglobin is a sigmoidal curve. So what is the reasoning for this? So myoglobin is 1 subunit, while hemoglobin is 4 subunits. This represents a tetramer for hemoglobin, and this leads to something called cooperativity or allosteric regulation. So what this means is that binding of oxygen to 1 subunit of hemoglobin increases the binding ability of oxygen at other subunits. So binding O2 to 1 subunit increases binding at other subunit. Okay. And we can describe this transition as being a transition from the T state to the R state, right? So the T state, this is low binding affinity. While the R state over here represents high binding affinity for oxygen. So essentially, as more oxygen binds to these hemoglobin subunits, it's transitioning from a T state to an R state. So when there's no oxygen bound, it has a low binding affinity for oxygen. which is why the curve slowly starts to pick up over here. It has a low binding affinity at the start, but then as more oxygen binds, as the partial pressure increases, it starts to transition to this R state, which has a higher binding affinity for oxygen. So then we sort of get into this exponential part, and then at high partial pressures, all 4 subunits have completely bound to oxygen, so it is completely saturated. And again, the P50 is the partial pressure at which there is 50% of that oxygen saturation. So 50% of all the proteins will have oxygen bound to them. It's very low for myoglobin because myoglobin only has 1 subunit, so there's no allosteric regulation. It just always has a high oxygen binding affinity. Hemoglobin, it takes a bit more because it initially has a low binding affinity. It takes a bit of oxygen binding to raise that to a higher binding affinity. Okay? So the next question, unlike hemoglobin, myoglobin is unaffected by 2,3-bisphosphoglycerate. Why? So we have to know what 2,3-bisphosphoglycerate does. So 2,3-BPG stabilizes the T state. So this is, remember, low binding affinity, okay? So low O2 binding affinity at the T state. So this is stabilizing the T state. So this is going to facilitate the release of oxygen to the cells. And this is because the 2,3-bisphosphoglycerate binding to the hemoglobin reduces the affinity of oxygen binding, meaning oxygen will be released from hemoglobin. So oxygen will be delivered to cells. Myoglobin is unaffected by this because the purpose of myoglobin, so myoglobin holds onto oxygen, right? So this represents storage. So storage specifically for the muscle cells. On the other hand, hemoglobin delivers oxygen. So it's picking up oxygen from your lungs and delivering it to your tissues. And because of this, it needs a mechanism to raise and lower oxygen binding affinity. So this is done through the allosteric regulation, switching between the T state and the R state, but it's also done through 2,3-bisphosphoglycerate. What that's doing is it's lowering the binding affinity of oxygen, allowing hemoglobin to release oxygen. So 2,3-BPG allows for release of O2 to the tissues, which is not something that myoglobin needs to do because it is used for oxygen storage. All right, for the next question, continuing with hemoglobin, beta thalassemia is a disease involving what protein subunit? So this is a disease of hemoglobin where there is not enough beta chain hemoglobin formed. So there isn't enough of the beta chain formed, so that means that there's too much alpha chain. So what this means is that the alpha chains accumulate as insoluble aggregates. So essentially, the alpha chains will sort of clump together. They will not be water soluble when they do that. Thus, that causes issues within cells and within your blood, okay? Because you have these big clumps of these alpha chains. So the next part is a patient with beta thalassemia lacks the gene for alpha hemoglobin, stabilizing protein, AHSP. Will their beta thalassemia be better or worse? So it will be worse. And this is because they lack the gene for alpha hemoglobin stabilizing protein. So we said beta thalassemia is a result of these alpha hemoglobin chains forming these aggregates. So this protein is going to stabilize the alpha helices so that they don't, or the alpha subunits, so that they don't form these aggregates. But without it, this means there are more aggregates. So AHSP prevents formation of alpha chain aggregates. So without this protein, without AHSP, more aggregates form. Right? So that makes it worse because there are more aggregates because you lack the protein needed to stabilize the aggregates. All right, the next question is about protein purification. So why do large proteins move through gel filtration quickly, and why do they move through SDS-PAH gels very slowly? So looking at large proteins here. So let's define what these two techniques are. So gel filtration is a type of protein purification. This is separation based off of size. These techniques are both separation based off of size. So here we have a column with porous beads. All right. And small compounds will enter these beads and get slowed down, while large compounds cannot enter the beads and they will move quickly. So small compounds or proteins enter beads and this slows them down. Meanwhile, large compounds, so large proteins, cannot enter the beads. So this is therefore fast. Okay. So that's much better. Slow and fast. Okay. So small compounds slow, large compounds fast. If we want to draw what this looks like, so imagine this is our column here. These are our porous beads. Okay. So the small proteins here will get caught in the bead and they will go slow. Meanwhile, the larger proteins, these cannot enter the beads, so these end up moving faster. All right. So SDS-PAH over here. This is also separation based off of protein size. This is using a polyacrylamide gel. Acrylamide gel. Okay. So here, the small molecules will navigate quickly and the large compounds will navigate the gel a lot slower. So small here will be fast and large compounds will be slow. So what is SDS-PAH? So SDS here will denature the protein. And this will give a uniform charge distribution. So essentially, it will untangle the protein and will give it negative charge all over, and that is proportional to the size of the protein, not necessarily to specific amino acid residues. So it essentially turns all proteins into a long chain, no secondary, tertiary structure, anything like that, and they're all have an even charge distribution. So that means the only thing that they share differently is their size, okay? So looking at what this looks like, the polyacrylamide gel matrix kind of looks like this. So the larger proteins back here, these are going to get stuck in the gel, so they are going to move a lot slower. Meanwhile, the small proteins can navigate the gel matrix a lot more easily, so they move a lot faster. So this is the direction of motion here, okay? And what this looks like in the gel is this is where you sort of enter your proteins up here, okay? And then down here we have the positive electrode, and up here we have the negative electrode. So the proteins will be negatively charged, so they will move from the negative electrode to the positive electrode. And what we see is we have a larger proteins will show a band that has not moved as far because they will not move as quickly through the gel, while the smaller proteins will show a band closer to the positive electrode because they will move a lot more quickly. So this is small, and this is large. So the difference is basically the beads here will capture the small proteins, while here the gel captures the large proteins. So the small ones move slowly with gel filtration, and the large ones move slowly with SDS page. All right, next up we have some more simplistic questions. So we are just defining things, and then we are doing true and false. So first up, define ELISA. So this just stands for enzyme linked immunosorbent assay, right? This is where we use antibodies to determine the presence of a protein, and we also can attach that to a fluorescent dye linked to an enzyme, and then we can relate the concentration of a specific protein that the antibody is binding to the concentration of that protein. So we can link concentration to color, essentially. So the enzyme produces color, and that color is proportional to the amount of protein in the solution. Next, define isoelectric point. So this is the pH where a protein has a net neutral charge. Okay, so at the isoelectric point, also called pi, it is where the protein will have an even balance of positive charges and negative charges that will perfectly cancel each other out, such that the protein will have a net charge of 0. So equal amounts of positive charges and negative charges. Okay. And then the next question is indicate whether true or false. So the first one is the fluorophore in GFP is on the outside of the protein. This is false, right? Because the fluorophore in GFP is actually on the inside of the protein. It's like a barrel-shaped protein with the fluorophore on the inside. Next one, extracrystallography works best with small disordered proteins. This is false, because it works best with small ordered proteins. So it works best with small proteins, and it works best with proteins that are ordered, not disordered. If it is disordered, it will be difficult to characterize the structure of a protein with X-ray crystallography. Next and last, NMR structures are determined from interactions between protons that are nearby in space. This is true. This is just the definition of NMR. It gives us a sort of graph that lets us know, gives us information on where the protons are in a protein, where the hydrogens are, based off their interactions within space. It can give us a lot of data that can help us map the structure of a protein. All right, this is the last question of the first practice midterm. So we are being asked to find the ratio between tris and tris HCl at the following pH values. So to do this, we need the Henderson-Hasselbalch equation, which is up here. pH equals pKa plus log A minus over HA. So the concentration of the conjugate base divided by the concentration of the acid. We also need to know the pKa of tris and tris HCl. This is given. It is 8.3. So in this case, tris HCl is HA because it has the H plus attached to it, while tris is A minus. Okay. In this case, it would probably be more accurate to say tris is A neutral and tris HCl is HA plus. That is the more proper notation for this. However, it shouldn't affect the problem too much. So in this case, at pH 8.3, so we are going to use pH equals pKa plus the log of the ratio. We just want to find the ratio. So here we get 8.3. Our pH is equal to the pKa 8.3 plus log of tris, which is A over HA plus. Okay? So now we can subtract 8.3 from both sides. You get 0 equals log A over HA plus. And then you can do 10 to the power of both sides. And what you get is 1 is equal to A over HA plus. All right, so the ratio there is 1. That means you have equivalent amounts of each. So the concentration of tris is equal to the concentration of tris HCl. Moving on, pH 9.3. So here we have pH 9.3 is equal to the pKa 8.3 plus the log of the ratio A over HA. And here we get 1. So 8.3 minus 9.3 minus 8.3 is 1 equals the log A over HA. All right, and then we can do 10 to the both sides, and we get 10 to the 1 is equal to 10 equals the ratio of A over HA plus. So if we get a number greater than 1 here for the ratio, that means we have more of the conjugate base than we do the acid. If that's the case, we expect the pH to be more basic than the pKa, which is what we observe, because at the pKa, when pH is equal to pKa, we have equivalent amounts of conjugate base and acid. When pH is greater than pKa, so more basic, we have more of the conjugate base than the acid, so that makes sense. Lastly, we are looking at a pH that is more acidic than the pKa, so 5.3 equals 8.3 plus log A over HA plus. All right, so we subtract 8.3 from both sides, and we get -3 is equal to log A over HA plus. Now when we do 10 to the minus 3, we get 0.001 is equal to A over HA plus. All right, so this means when we have a number less than 1 for our ratio, this means we have more acid than we do conjugate base. If we have more acid, we expect the pH to be lower than the pKa, which it is. It represents it's more acidic than at the pH where we have the equivalent amounts of both the acid and the conjugate base. All right, so last question here. Is tris HCl an effective buffer at pH 5.3? The answer to this is no. And that is because buffers work best when you have an equivalent amount of each. So buffers work best when the pH of the solution equals pKa. So best buffer is when pH is equal to pKa. Here 5.3 is about 3 off from the pKa, and in this case it means we have a lot more acid than we do base, about 1,000 times more acid. So we have more HA than the conjugate base A. We have more tris HCl than we do tris. So this will resist the addition of a base well because we have a lot of acid to counter the addition of a base. However, we do not have a lot of conjugate base to counter the addition of an acid. So this would not make a good buffer because this would resist addition of a base, but not of an acid. Versus at pH 8.3, which is where it would make the best buffer, we have equivalent amounts of acid and conjugate base, meaning if we add an acid, we have enough conjugate base to neutralize it. If we add a base, we have enough acid to neutralize it. And that is it for the first practice exam. So yeah, see you next time for another practice exam much like this, going over different questions. Thank you.